Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor and your host. On DNA Today, we spend a lot of time talking about genetic testing in the United States. But what does this look like in other parts of the world? In this episode, we're shifting our focus to Italy. And since it's Breast Cancer Awareness Month, being October, we're discussing breast cancer genetic testing. To learn more about this, we're joined by two wonderful experts. Dr. Beatrice Salvatori is a scientist and project manager at Negedia, where she leads oncology-related projects and develops new research initiatives. With a PhD in genetics and molecular biology, her career has spanned bioinformatics, breast cancer research, and now applying cutting-edge sequencing to expand access to hereditary cancer testing in Italy. Dr. Javier Batista Perez is a DCS specialist at MGI Tech. For those that don't know, DCS stands for DNA sequencing, cell omics, and spatial omics, with expertise of how MGI Tech sequencing technologies are being applied across Europe and how they're shaping the future of precision medicine. So we're going to get into how this initiative is reshaping cancer genetics in Italy and what lessons it might hold for global care. Thank you both so much for coming on the show all the way from Italy.
Thank you for inviting.
Thank you, Kira. It's a pleasure to be here today.
So, Dr. Salvatori, I figured we could start with you and giving a very basic overview and orienting people to what we're talking about today with hereditary cancer. So, how much of cancer is caused by lifestyle and environmental factors versus this smaller part that is caused by hereditary cancer?
Yeah, sure. So at its core, cancer is genetic. In the majority of cases, mutation arise in the tumor cells, often triggered by environmental factors such as UV, processed food, chemicals, and so on, smoking. But sometimes even people who have a very healthy style develop cancer. And when this happens, especially at a younger age, it is very likely that the nature of the disease is of hereditary origin. This means that we may inherit from our family variants in genes that may increase the probability of developing a tumor. Variants in genes, like genes involved in DNA repair, can make it easier for additional mutations to build up, especially mutations that could be advantageous for uncontrolled proliferation and so leading to a tumor. And since we're talking about breast cancer is a clear example of that. It's one of the most frequent cancer in women, about 25%, representing about 25% of all other tumor types. And in Italy, specifically, 55,000 people are diagnosed every year with this type of disease. And at least 10% of these people, so we're talking about 6,000 people, are hereditary of the disease. So we're talking about pretty relevant numbers.
Yeah, and it's important that you're highlighting this difference between hereditary cancer and, I would say, the other forms of cancer like environmental and familial cancer. So looking at breast cancer, as you were highlighting, because breast cancer is so prevalent, you're talking about like 55,000 people diagnosed in Italy. I think about another stat of 1 in 8 females being diagnosed with breast cancer in their life. I think that's a more global statistic. I'll fact check that in the notes. So why is this genetic testing for breast cancer, hereditary breast cancer, so important for Italy's public health system? What are your thoughts on that?
Well, this is true for Italy, but also for other countries. It's a global issue. But in Italy, as in many other parts of the world, the focus always has been on catching symptoms early in order to manage better the patient and don't get a disease that it's already too aggressive. And mammography is the main tool offered through the public health system. In Italy, at set ages, depending on the population risk, we have from 50 to 69 years old women that are entitled for a free screening every 2 years. And that has been demonstrated that has improved breast cancer management by decreasing mortality around 30%. So numbers are pretty convincing that preventive or just the screening is very helpful. But technology is moving forward, and now we have a new tool that is next generation sequencing that has been in our hands, at least in the scientific research hands, for around 20 years right now. And I think we are at the moment that this tool may be actually very helpful also for the clinical world. The next generation sequencing is the most powerful tool to look at the genetic information. So understanding if a person is a carrier of a variant in one of the critical genes that are associated with hereditary form of cancer may lead to early prevention and best monitoring. So this could help us, for instance, decide who might need the screening at an early age and who may not, or even who should perform preventive surgery. And ideally, we can identify those people even before the symptoms appear. And this is really important.
Yeah, it does provide that opportunity if a gene mutation or pathogenic variant is found in one of the genes that we'll talk about related to hereditary breast and ovarian cancer syndrome, then screening can start earlier. And so possibly that cancer can be detected much earlier so that it's at an earlier stage, it's easier to treat, and all of those downstream effects. I want to get into this genetic testing because you're bringing up the sequencing technology of next generation sequencing. And I wanted to talk about what the genes are that are included. So the genes I think about is BRCA1 and BRCA2 of being the first two discovered by Dr. Mary Claire King and her team back in the 90s, but we found more since. So if someone is doing this testing, what genes are included? Are we stopping at BRCA1 and 2? Or are we including some others at this point?
Sure. So yeah, you're right. The routine genes that most of the clinicians use are BRCA1 and BRCA2, and they're actually the best known actionable genes and that are associated with the majority of hereditary. We talk about breast cancer and ovarian cancer too, covering around 10% of the cases. But we know that genetic doesn't stop there. We have other 20,000 genes. Obviously, they're not all of them relevant, but today it's known that around 100, 150 genes, a little bit less, are associated with hereditary forms of tumors. Then, if we just focus on breast and ovarian cancer, we can shrink this number to these other three genes that are very relevant and are CHEK2, PALB2, and ATM. And as I will maybe tell you later, these three genes are very relevant in terms of what they do inside our cells, but we also find them in around 2%, 1% of the hereditary tumors. So they still represent an important number of people where they could actually, once detected, could help prevention of the tumor symptoms.
And so how did you and your team end up deciding these 5 genes? It makes sense, BRCA1 and 2. I think most people are familiar with that, especially listen to the show here. How did you decide about the other 3 genes of CHEK2, PALB2, and ATM? Are these genes the top 5 in terms of frequency that are found for hereditary breast ovarian cancer syndrome? How did that come to be?
They are actually the top 5 genes mostly associated with hereditary forms of breast and ovarian cancer. Obviously, they're also associated with other types of cancer like prostate, pancreatic, So it's not only breast and ovary, but we selected them based on these two tumors. And obviously, our choice was totally based on published studies on what is it's known from the clinical community. And this is how we decided to focus on these 5 genes and not more.
Yes, you have to say, all right, well, we want to include as much as we can that makes sense and that's going to be effective, which we're going to also talk about a little bit later. Before we move on to that, though, I'd love to dive a wee bit deeper into how mutations in these 5 genes do contribute to a higher risk for breast, and we're also talking about ovarian cancer and some of these other cancers, but how are they related? Are any of the genes related to BRCA, like out of those other 3 genes?
Yes, they are. So especially CHEK2, but also PALB2, they are associated with the molecular function of BRCA2 specifically. So these genes are genes involved in DNA repair, so are the one that should control that nothing wrong happens during replication, during the life of our cells. What happens is that if we have variants in these genes that impair their functional activity, something wrong may happen, and our cell maybe not always be as good as the normal as a cell to repair what went wrong. And that's how happens that people that carry these variants are more likely to have a tumor rather than people that do not carry these variants.
Yeah, that's a good way of explaining it, that these genes protect us from developing cancer. So if the gene is broken because of a mutation, that protection is down and cancer can proliferate.
Exactly. It still works, but it's not working at the best. So we have to monitor ourselves a little bit more often in order to make sure that nothing wrong is happening.
And Dr. Batista Perez, we got to bring in your expertise now, especially coming from the industry perspective. Why do you think MGI Tech is such a great partner for this project and developing this panel and getting a bit into the technology that makes MGI Tech innovative and such a good match?
Thank you. So I think this is from the very beginning when we started in contact with Negedia. The thing that they were looking for, right, is the NGS technique that will be, let's say, very accurate, because when we're talking about this panel, the mutation, we're talking about even a single mutation. So we change just a letter on the DNA code and then everything changed. And then you need to have the power to look with this level of accuracy to find variants that have only one single nucleotide mutation. But at the same time, you want to link it to, let's say, the public health system in Italy. You need to make it in the way that it is affordable and in the way that you know more or less what are the time around time. So when you're getting the results. And then it was a natural fit for MGI platform. And then when we're working with Negedia from the beginning, it's not like we are instrument sellers, right? So we are a partner. I think this is the philosophy of MGI, and that's really helped everything since we started. So Negedia was probably the first to have a T7 sequencer in Italy, and also is the first certified stomics service provider in Europe. So then you can really see how is the good relation that we have and how we, from the beginning, right, find out that we are the right partners for not only these panels that we are talking here, but for many other applications that we are also working, and then having in the same institution, on the same roof, the capability of doing NGS at the level they can do, and also doing the special transcriptomics, definitely put Negedia and MGI partnership in the great shape to target it, all the type of diseases and research. I think it's also combining the research and the clinical aspect together under the same roof what make it special.
Yeah, I think that's really good points of just being able to utilize this technology And we're going to talk about just the details of why that's so great. And part of what Dr. Salvatori was bringing up is that we're looking at these, for this specific panel, we're looking at these 5 relevant genes to breast cancer. And an aspect that's changed over the years is looking at just some hotspots on the gene of like a few different known mutations versus sequencing and reading through the entire gene. Or if we get a little more into the details, maybe just the exons or the active parts of the gene. So I'd love to get into how that is with this. Are we looking at just exons? Are we looking at exons and some non-coding regions so that we get the most complete genetic picture that we can, as opposed to what we were doing years ago of like, oh, let's just look at the common spots that have mutations?
I can take this answer if you'd like. So the design was made in collaboration with MGI and with the researchers at Fondazione Teleton. And the design was with the goal, the aim was to cover all the exons. So we covered the complete coding frame of the genes, but we also wanted to span through the untranslated regions that are at the 5' and 3' of the RNA. And not only that, we also added those splicing and regulatory regions that are very important for a gene to form and be mature correctly. So we are not including just those hotspots, but we're including the full frame of the genes. And we are sure that we're covering basically almost of the known variants associated with hereditary mutations in these genes.
I, I would like to add that when we are talking about the non-coded regions, that's sometimes are the more challenging one to sequence with the high accuracy, especially in the region that are GC rich content. And then having the platform, the whole design is doing collaboration, but it's owned by Negedia, but powered by the T7 sequencer they have that allow them to do it in the high throughput and high accuracy, so they can actually look at the single nucleotide mutation, even in this non-coding area, where this is more challenging. So I just want to add this part.
Yeah, that's so valuable. And I'm thinking just from a bit of a historical perspective, the reason that we started with those hot spots is because we knew, oh, if it's this change, this is what that means. Whereas when you start looking at all the exons and spanning out a little bit more, as Dr. Salvatori was saying, all right, well, let's span out and zoom out a little bit from the exons to see what's on the ends of those, that now we're going to find a lot of variants or changes that we're not sure what that means. And nowadays we have databases that are growing so that we understand, or we have a better chance at understanding, okay, we see this genetic change. Let's go to the database. Have we seen this before? How many times? Does this mean that it affects the gene, that it's not going to work, that cancer protection is not going to be there, or at least not be in full force like we want it to? So I'm wondering how these databases between Negedia and MGI of just being able to curate variants and how that's gone, especially for these 5 genes that we're focused on, because they are common genes. So I would hope that a lot of the variants picking up, we are able to curate, and we're still going to have some VUSs, but yeah, not sure any thoughts on that.
I think that the database is really getting improved, right? So you're looking at databases like ClinVar or Genome ID. They are definitely getting better and better, and they are covering more the, let's say, population from Southern Europe, where Italy it is. So it is easier to actually cache in the most of the variants and don't get in the situation that you mentioned, that you have a variant that you don't know how to annotate. And we also have other AI tools that are getting really helpful for the annotation part of the variants. But the problem here is that if the sequencing is not like rock good, right, then it will be very difficult to use the data. So all these databases are good. The AI tools to do the annotation are good, but if you don't have a good data to start with, then the outcome is not very, you get not very confident, right? You might have like a false negative. You might have some things due to the lacking of accuracy of the sequencing itself. So I think in this part is where, having the MGI technology that is getting also improved, which the time allows the Negedia in this case to have a good rock solid sequencing data that then they can go in and and using the database available to do the annotation and then have the final report. So, but I let to Beatrice if you want to add anything.
No, it's totally correct. It's true. We're covering the whole coding frame and also the other regions of the RNA, but we are still focusing on genes that are very well known, and especially BRCA1 and BRCA2 are commonly sequenced, maybe not for the full frame, not for the full length as we are doing, but we already have a lot of information from these genes, and a lot of studies that can help us to understand and interpret the variant that we find.
I think even when you have increasing your database, like you even own databases, as you're going on with the test, might even find the correlation between the different genes. What are the level of expression, the mutation, and then it might help you to subdivide in different subgroups. That then you might have a different therapeutic approach or prevention approach. So I think that's something that definitely can be super helpful as a database on Negedia, but globally also growing and with the use of the AI. Always having a rock solid sequencing data.
It's also very helpful that you're doing a lot of sequencing of a more specific population of being in Italy, as opposed to other places that don't have that data set of this certain population, because we see that as a problem in a lot of databases of it's overrepresented of some ancestry of some areas of the world and underrepresented in a lot of others. So I imagine that's also very helpful for people that do have Italian ancestry to say, oh, well, do testing at Negedia because you have that database of variants that are more likely to come up for them as opposed to people with ancestry from other countries.
It is true, but in Italy, we're Caucasian, like most of the US and Europe. So we don't have a very close, except for Sardinia, some areas of the islands that have been very inbred in the last century. But Italian population is Caucasian and shares most of the variant with the rest of Europe and also with the US. That it's, as you know, we have many people that moved from Italy to the US and now are US people. So I think our DNA is pretty common also with other countries and other parts of the world.
Yeah, I totally agree. I think, but in my point of view, I think the Italian genome is very diverse. I think it even is similar. We are mostly Caucasian. I think because of the Roman Empire, I don't know whatever reason it is really mixed. And then you can find representation of many other regions. We can find the part of it in the Italian genome. So that also could be very attractive. So I think whatever we are actually developing that works for Italy have a great chance that will work for many other parts in the world, right? So this is, I think, a positive point as well here.
And Dr. Batista Perez, I'd love to learn more about how MGI's technology is making this panel. We've talked about how it's more accurate. Let's talk about how it's cost effective, because people love hearing about savings. So how is this cost effective of using this technology from MGI Tech as opposed to other more traditional sequencing approaches and maybe what other companies have available? Because making it more affordable makes it easier to integrate into Italy's public healthcare system and then helping a lot more people.
Yeah, thank you. So I think this, let's start with the more accurate, because we didn't go actually into detail why this actually is have the accuracy come from. So this is our DNB-seq technology. So it is using the 529 enzyme. So we're starting with the DNA template, and then we are doing before loading into the flow cell, we are doing like we call DNA nanoballs. So starting to get the template and do a copy, and then they displace this copy, and then they do another copy. They make like very long single strand DNA, and when it gets long enough, it starts to entangle and make these balls, right? That we call DNA nanoballs. But because we do that before loading into the flow cell, we don't have any chance to having like 2 of those DNA nanoballs in the array that we have in our flow cell. So duplication rate is very, very low. What means you get more data. That also goes to saving money, right? Because you get more data from the single flow cell. But not only that, because you do this DNA nanoball from the same template, there is no any exponential expansion of the error. So you have 1 error, you might see it in one copy, but because you are doing it right, uh, many copies, then at the end you can correct the error. It is very difficult that you will scale this. So it is very, very important when you are doing these panels, but we are looking at the single mutation. And then on top of that, looking more about why it is more affordable, I have to say that it's very flexible. So it have a, first of all, very high throughput. So you can run many samples at the same time in the one single flow cell, but the T7 is like 4 sequences in one. So you have like 4 different individual small sequencers, let's say, where you can sequence there all at the same time, or you can sequence one, and you can sequence 2, 3, or even you can do different application on a different compartment of the sequencer. But this really allows to have the sequencer up and running all the time, so you don't need to wait. So you have like you can have more. You will know what is the turnaround time. So it's not like it depends if I get the sample, if I don't get it, because you can actually do one flow cell, you can do 2, or you can do more, right? And then this definitely will decrease the cost because you are able to do the sequencing for many patients at the same time. So, and my belief is that with MGI technology, that they're getting more and more innovative, because that's MGI innovation, the cost will decrease. So now we are all into having a cost that will be, as expensive as having MRI or having like a TAC. So at the end, when you're getting at this level for doing the sequence of the whole genome, then you can imagine what you can do when you just sequence a part of it, when you're doing it targeted.
And when it comes to people actually being able to access this and have this testing ordered so that all of this technology can be happening in the lab to get these results, in Italy, do general practitioners play a role in ordering testing or referring patients to a specialist? How does this work in Italy? Because I'm not as familiar as being someone that practices in the United States.
It still works like the United States and the rest of the world, I guess. The genetic test should always come with a consultation by a geneticist, and that will explain what the test is for, what the limits of the test are, and also will help the patient to understand the results once the patient gets the results. And only in this context it can be available, because general practitioners aren't prepared right now to handle something that is still part of the genetic field. So also the oncologist, sometimes even when patients have to take these tests, once sometimes they already have the disease, but they want to take the test because they want to know if they have an hereditary form of cancer, maybe to inform also the other members of the family. But also in that case, the oncologist suggests the patient to go to a geneticist. So this is really important. So all the part of the consultation is still very relevant.
Yeah, that's helpful. And I'm sure it will change in the future of just how ubiquitous genetic testing is becoming, that we're going to have some testing probably be done by general practitioners. But for now, that's certainly like the best way to do it, and pretty similar to the United States. I mean, there's some GPs that might order certain testing, but it is a little bit dicey of, do they really know what they're ordering? Are they ordering the right test? And it sounds like from what we've talked about, the people that are getting this testing have some kind of family history or a personal history that is leading them to do this hereditary cancer testing. But I'm wondering if both of you predict that this could change in the future, where Italy and other countries move towards a population-wide genetic testing of these 5 genes, because sometimes people don't have their family history for various reasons, or there just isn't a history of that, and it turns out they still do have one of these pathogenic variants in one of these 5 genes. So what are your thoughts on moving towards population-wide testing?
So this is a very good question, and it's actually, we can answer these questions only with data and the proof that doing a wide screening may help the monitoring and the catching earlier the symptoms of the disease. So as it was shown for the mammography, that was able to reduce by 30% the number of deaths. What we are doing right now, and we're doing this in collaboration with Fondazione Teleton, that has a research center that always collaborates with us. We're trying to demonstrate through a clinical study that a screening on healthy people through these 5 genes may help their knowledge of the type of may help clinicians to symptoms of the disease even before they get harder to cure. So it's the same concept of mammography. That's basically what they will do, unless obviously they are found to be carrier of a very pathogenic mutation in these genes. They will do more monitoring at an early age, and that should help the cure of the disease. So we need to have those numbers in order to convince all the clinicians and the rest of the world that these screenings are really helpful. So it's just going to be the numbers that will demonstrate what we are saying right now.
I've never thought about it that way of everybody gets mammograms and should, that are assigned female at birth, have that breast tissue. But I've never thought about doing population genetic testing like another step in the evolution of being able to prevent breast cancer. I love that concept of saying, hey, we're doing this with mammograms. It's sort of the same vein there.
It's not this, we do the genetic testing first, and then if you have a higher risk, you just don't start to do the mammogram at 50, but you start earlier. And this is the only difference. So you have the knowledge that you have to be screened more often and earlier in life. So this is just what changes. We don't have to be too worried, so this is why we always need a consultation first from an MD, from a doctor. But we just need to understand that having this knowledge empower us to a better prevention.
I think, that it's not only that the level of detection is what you can learn. What if we find for this healthy population another, group of people that they have different variants, that they more related to the stress? Maybe you can find like some hotspot when you see the incidence of the breast cancer is higher, but you cannot link it to the hereditary problem. So that will be very interesting to see what you can get there, right? Maybe there is a place that they have like a lot of stress because of contamination. It's a specific type of pollution or lifestyle there, or whatever reason they can actually contribute to the generation with different type of mutation that now you can find. Maybe you cannot call it the first time because they are not in the database, but then we're adding those to the database. So I think this is definitely a good thing, and we are moving in that direction. What is the problem here? First of all, we need to have a good accurate data. Then we need to decrease the cost of the sequences, because otherwise it's difficult to make it massive for everyone. And then at the same time, we need to improve how we interpret the data, because now NGS, and not only NGS, are generating enormous amount of data. And honestly, some of the data scientists, they are very struggle how to manage all those big data and how we can get the best out from those big data. So these two things actually can improve, right? You decrease the cost, you increase the interpretation tools to get it faster, and then we can have a gradual expansion. I see it more like, maybe starting for hotspots, maybe for a CT, and then definitely you can later scale it up to the bigger context. For doing that, you need to have the infrastructure. And that's also the problem. It's not so easy to make this population genomics project, because you need to have a lot of sequencer with high throughput. And then I think in this sense, MGI is really working into it. So we have a giant that is a team. And, right now, this is not many people that actually can feel, because it's hungry monster to have the level of processability that he can get. But when we are getting there, I think this will be the type of sequences that we need, right? That when you can actually have a hubs in the country and you can just send samples for different areas to the same hub, and then they get sequenced all together to decrease costs and improve the turnaround time as well.
All that needs to happen to make it scalable so we can even get to the point. You're right, Dr. Batista Perez, of being able to say, okay, if we want the whole population to be routinely doing hereditary breast cancer genetic testing, yet we need to make it feasible to do that. And scalability is such a big part. Dr. Salvatori, any other thoughts on other challenges or just the opportunity to have it be population-based and scaling it of, we can even span out just from Italy, but globally of other, probably starting with developed countries?
Well, first of all, we have to create we have to be aware that knowing all this information obviously opens a lot of questions, but we don't have to create unnecessary alarm. So the first thing is that we need to move also the next generation of doctors and clinicians closer to precision medicine. And the next generation sequencing is the tool of precision medicine. And so we have to get a little bit closer to these clinicians, making them understand that obviously having all this information is complex, but we can handle it. Just this panel that we are bringing here today, it's an example. So we're not focusing on all the genes, we're just focusing on 5 genes. So we don't want to give too much information. We just want to focus on those that are clinically relevant. But at the same time, we want to give a new tool for prevention, for improvement of the management of this disease. And if I can add something, it is true that the sequencing goes with very high throughput technology, but also this small panel that we are talking today also goes a smaller sequencer. So clinicians that usually have this sequencer and use this sequencer in their molecular pathology lab, in their laboratory, genetic laboratories in the hospitals, may handle the use of these type of tool and technology also in their hands. It's not so complicated. Obviously, Negedia has the best and has the most processive sequencer, and this is how we always set up the technology. But I know also MGI has sequencers that are able to go in smaller labs in the clinical world, and this type of kit can be transferred also to those realities. So it's not something that only Negedia can do. I think it's something that can be transferred also to the clinical world.
I think this is a very good point. This is what we really want to do, right? Is enable to the clinician to have the right sequencer for the right use. So, for example, you are working in in intensive care. You need to have an answer like really fast. You don't want to wait to send your sample to Nigeria, or you don't want to wait even like 3-4 days to have the sequencing done, or you don't want to pay a lot of money for doing this because they're not affordable for the health system. So we have different throughput sequencer, as Beatrice say. And then there are, for example, one, the G99, that it really can do the sequencing really quick within 12 hours. And then you can use even pull different patients together in the flow cell. So we have different throughput, maybe a little bit more expensive that you do it in Negedia, but you can do it in the local hospital and have the result the next day. So this is definitely very, very helpful. But I've been noticing, like in Italy, one big problem to make this a reality and make the panels available for the whole healthcare is that they actually need to go into the insurance so people can get reimbursed for doing the test. And then there is I think it's now a gap between the geneticists in Italy and the clinicians and then the Minister of Health. So there is no good communication. So everyone more or less agree that it's good, but at the end of the day, right, there is no this alignment between the 3 parts to make it more efficient and faster to get this type of panels in the insurance, so everyone will do it. So it's not the same if they tell me it's good for you to do that, but you need to pay €300, and it's good for you to do that, and it's free. The government cover the test. I think this is also the things now that having places like Nigeria, and then the clinician, the Minister of Health see that this can be done that fast at that cost with this accuracy, then might have to one day eventually disease can get more spread in
one step at a time, right? To be able to make that progress. And I really like how you both highlighted that MGI Tech has so many different tools, and every lab or different hospitals or different situation may say, well, this tool is going to be better than that one for us for these different reasons. So I think just the versatility is really helpful there. And I'm just excited to see how things progress. I think cancer moves so quickly in terms of the research and development that's happening. And then genetics moves so fast. So you put the two of them together. It's like hard to keep up with everything in the best way because of all the progress being made. Thank you so much, Dr. Salvatori, Dr. Batista Perez, for coming on and sharing all of your expertise within hereditary breast cancer genetic testing, and then also with the lens of in Italy. I learned so much, and I'm sure the listeners did along with me. So thank you both for coming on.
Thank you.
Thank you.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.